Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. I am Professor Adam Ahmed Adam. You are welcome to this bacteriology lecture. It is about introduction to bacteria. The word bacteria is plural. The singular is bacterium. Bacteria are unicellular microorganisms. They are prokaryotes. The bacterial size, the length ranges from 1 to 3 micrometers and the width ranges from 0.25 to 1 micrometer. Bacteria are dense population in Earth. They are more than all plants and animals. There are about 5 times 10 to the power 30 bacteria on Earth. They constitute about 13% of Earth biomass, the living organism, versus humans. Humans constitute about 0.01% of Earth's biomass. In soil, there are about 40 times 10 to the power 6 bacterial cells per gram of soil. In fresh water, there are about 10 to the power 6 bacterial cells per ml of fresh water. Classification of bacteria. They are classified according to their morphology, their shapes, and their stainability to gram stain. Regarding the morphology, the bacteria can be cocci, which are rounded, circular. The singular is coccus. They can be bacilli, the singular is bacillus, as rods or sticks, and coccobacilli, singular coccobacillus. Here the shape is in between a coccus and a bacillus. The spirochete, here the shape is a zigzagging shape like what you see in the picture. We come to the stainability by gram stain. So they are either stained by gram stain or not stained. The stainable bacteria are either gram positive or gram negative. The gram positive bacteria, they appear brown because they retain gram iodine, while the gram negative bacteria can be, or they appear red because they do not retain gram iodine. The gram positive can be cocci and bacilli, but the gram negative can be cocci, bacilli, and coccobacilli. The spores. Some bacteria produce spores, some of them, not all. The spores are resistant stages against adverse physical and chemical conditions and nutrient deficiency. These are adverse conditions. When the vegetative bacterium is exposed to adverse conditions, so as not to die, it changes itself into spore. But when the conditions become favorable, the spore will germinate into vegetative bacterium, as you see in the figure. The comparison of gram positive to gram negative. The upper one gram positive, the lower one is gram negative. You see here the black arrows are pointing to different structural components, but the most important, the red arrows. Red arrows are pointing to a component peptidoglycan in the cell wall. It is thick in gram positive but thin in gram negative. So being thick in gram positive makes it gram positive. Look down in gram negative, the outer membrane, the green arrow. This is the lipopolysaccharide plus protein, endotoxin. This is a virulent factor. It is found in gram negative but absent in gram positive. The genetic material. Chromosomal DNA contains a single chromosome. This is found in all bacteria. It is circular, double-stranded DNA located in the nucleoid. There is no true nucleus in bacteria. Plus or minus plasmid. Plasmid is an extra chromosomal DNA found in some bacteria, not all of them. Look at the figure. This figure illustrates the different components of a bacterial cell with their function. But we have to notice that the asterisks are pointing to areas where are not common to all bacteria, found in some bacteria only. In the extreme left-hand side, the flagellum. They can be many flagella. Flagellum is an organ of movement. It is found in some bacteria. You go to the right, the pilus. There may be pili, very many of them. The pilus is organ of attachment found in some bacteria. Plasmid carries antibiotic resistance and virulence, the ability to cause diseases in some bacteria. The capsule, which is a virulence factor, it is antiphagocytic, it resists phagocytosis, it is found in some bacteria. The mesosomes, to increase the surface area in some bacteria. Lower down, the photosynthetic membranes in some bacteria. The rest are common to all. The red color illustrates the function. The black writing illustrates the organ or the component. Gene transfer between bacteria. One vertical gene transfer, two horizontal gene transfer. In the vertical gene transfer, this is by binary fission, division. The mother bacterium divides itself into two daughter bacteria. Here, the horizontal occurs through one of the four ways: conjugation, transformation, transduction, and visiduction. Let us take them one by one. Conjugation. In conjugation, a sex pilus forms a tube between the donor bacterium and the recipient bacterium, as you see in the picture. The donor bacterium produces a copy of the gene to be transferred and transfers it to the recipient bacterium through the tube. And then the tube closes. Transformation. This is carried out by competent bacterium. If the bacterium is exposed to stress, it will be competent. How? It will be able to take a gene in the environment, free gene in nature, in a gene pool. So the bacterium becomes competent and uptakes free DNA from a gene pool. Transduction. The DNA is taken from the donor bacterium by a bacteriophage. Bacteriophage is a virus that infects bacteria. Then the bacteriophage injects the genes into the recipient bacterium, as you see here in the picture. Vesiduction. The donor bacterium forms an external vesicle containing genes, maybe one or more, and attaches itself to the recipient bacterium. Then it internalizes the DNA, as you see in the picture. So what is the importance of horizontal gene transfer? The transferred genes, the DNA, transfer the properties of virulence, the ability to cause disease, from a pathogenic bacterium to non-pathogenic bacterium. So the non-pathogenic bacterium, after receiving the genes or the gene, it becomes pathogenic. And resistant to antibiotics, the ability to resist antibiotics, also this property is gained by the horizontal gene transfer. By this, the lecture is over. I hope that it is understandable, and I wish you all the best.